Okay, back to continue now with the post-DNA video, post-DNA structure presentation, right? So let's get into that particular slide. I do want to spend a little bit of time with the analysis of the whole process, right? So a big misconception a lot of students have is that everything that we know today has like always been known. Oh yeah, DNA, yeah, I know what that looks like. We've always known what DNA looks like. We've always known what DNA does, right? That's a big misconception. And again, this date, February 28th, 1953, was so very significant, right? If you watched the video, you saw that up until that day, it was thought that proteins were what convey genetic information, right? If I've always wanted to get a biology book that was published before 1953. I think it would be quite interesting to analyze. It would be all wrong. It would be completely incorrect, but I think that would have been interesting to see what they believed was correct and what they believed was fact at that time, right? So that proteins were that macromolecule that conveyed genetic information, right? And that's false now, right? And we know now that DNA is that molecule. And we know now, after 1953, of that anti-parallel double helix, complementary adenine with thymine, guanine with cytosine. That is what Watson and Crick gave to the scientific community, and Nobel Prize type of discovery. So I do hope that you saw their collaboration, right? And I do hope that as you continue your academic careers, I think it's a little challenging right now. We can maybe try to start improving on this a little bit, but you can learn a lot from an instructor, but you can learn a lot also from peers. And that give and take of information was something that really enabled them, that similar drive, that similar goal that they wanted to achieve. That is what we consider very good collaboration, right? And collaboration has the potential to carry you far in this academic career, right? So I do hope you strive for that. Competition is something that is that driving force, right? So here we introduce Linus Pauling, right? Pauling is going to represent a lot of people around you, right? And there's going to be some days that I'll get to it tomorrow. I'll get to it later. But there's some people that have a little bit maybe higher drive than you, and they're not going to wait till tomorrow. They're going to get that done today. And they're out there like, man, if it wasn't for that person, I could sleep late. I could relax this week. But no, I got to work hard because they're working hard. So that is that driving, competitive sort of force that Pauling was, right? So Pauling, that sort of friendly competition with Watson and Crick, and they both kind of that competitive energy, that competitive back of your mind, that they're doing stuff, that entity that's there. I think that was an important role for both of these labs, yeah. And Watson and Crick got there first. They discovered it first. But Pauling had an indirect motivating force in Watson and Crick's sort of arrival. Now we look at maybe a poor collaborative effort. Wilkins and Franklin, difference in personality, difference in maybe egos. As they say in the video, in 1952, 1953, you can imagine how difficult it would have been for a woman to be taken seriously in the field of science, right? Even today, in the year 2020, females are still underrepresented in the scientific community. So we commend Franklin for doing what she did. Who took that famous picture, right? If you saw the video, that famous photo 51, that picture, that double helical radiograph there was generated by Franklin. And this has been I guess some conflict, some unresolved bad feelings for a long time, that if this had been today, I firmly believe that Franklin would have been included in that Nobel Prize discovery, right? Being the time, being the cultural state that was in 1953, she was excluded from the Nobel Prize. And again, working with all this radiation, and really had a very bad impact on her health. She sort of died with not receiving the recognition that she probably should have. But again, that was the way it worked out at that time. And again, probably for the not fair it was not fairly acknowledged, the contribution that she gave to science and to the discovery of the DNA molecule. So yeah, so I think this was very, very relevant. Yeah. So there's Franklin and her famous photo 51 there. So the moral of the story, again, we don't always know how to do stuff. We don't always know how to answer scientific questions. How do we come up with a vaccine for the COVID? How do we prevent COVID? Is COVID going to continue to evolve? Well, we don't know, right? We apply the scientific methodology. We're going to get it wrong sometimes, but eventually we will get it right, right? And that's how science, that's how our basis of knowledge grows, by the application, the process, the step-by-step process of conducting the scientific method. And again, always strive for collaboration. Use the competitiveness in you and the competitiveness around you to kind of keep that driving force and moving forward, right? So that's the goal. So with that sort of summarizes the video that I wanted you to watch. Now we get into the application, right? This is something that has revolutionized forensic science. If we were forensic scientists in 1951, 1952, we would look for fabric fibers. We would look for hair fibers, maybe for fingerprints. We would look for footprints in the mud, blood, that kind of stuff. Well, in the year now, 2020, we can do a lot with very, very minimal amounts of information. All we need is a little cell, a little drop of blood that we can extract DNA. And once we extract DNA, we can then come up with the entire genetic instruction. We can come up with the basically what an individual looks like based on their A's and C's and T's and G's. So quite amazing. So let me give you an example here, right? So over here, we're looking at the A's and T's and C's and G's. The DNA molecules are going this way, or I should say this way. Well, if this thing starts to shift over and is going this way, so if our sugar phosphate backbones, if our ladder isn't upright, but if our ladder topples over, right? So we're now going 5 to 3. So we got to be going 3 to 5, this direction. So 5 to 3 this way, 5 to 3 coming back this way. So we're looking not at a vertical analysis, we're doing it horizontal because it's just easier for me to type this way than vertically here, right? But let's say that there was an accident, there was a crime scene. Ooh, yellow tape is never good. Chalk outline, ooh, somebody's day didn't go as planned. The forensics team got there, let's say, a little bit late. And it's been hot lately. Maybe that blood started to decompose. Bacteria started to decompose those cells, that DNA. But the forensics team got there. They did what they could. So let's say that we know there's going to be a sugar, a phosphate, a sugar, and a base. We know, right? Because we know DNA. We know it's a repetitive sugar, phosphate, base, sugar, phosphate, base, sugar, phosphate, base. But they're not going to show sugar phosphate because it's boring and it's repetitive and everybody knows that it's sugar phosphate. Over here, another sugar phosphate, sugar phosphate. The diagnostic part of DNA is the base sequence, the particular sequence that we can arrange the A's and T's and C's and G's. If you know something about computers, the computer language is a series of binary, right? It's just a 0 and 1. And if you know a little bit about binary code, you can do a lot with computers. Computers do all kinds of stuff, and they're only operating on an alphabet of 2 numbers, right? 0 and 1. And depending on how many zeros and ones we have, the arrangement of the zeros and ones, it makes that alphabet quite significant with only 2 letters. Well, imagine how much more complex that instruction can be if we have 4 combinations, right? So we have A's and T's and C's and G's. So it's not binary, it's like a quadronary, something like that, right? Quaternary almost. So four potential codes, and imagine we have a million of these, DNA strand with a million of these A's and T's and C's and G's. How many different ways can we adjust, combine, alter that sequence? It's almost mind-blowing how big the diversity is of the DNA molecule. Just think, in your lifetime, have you seen 100,000 people, 100,000 faces, right? Well, yeah, you might have seen twins, but for the most part, almost everybody has their own different composition. We all have 2 eyes, 2 ears, a nose, and a mouth, but there's different compositions, different thicknesses, different features, and all that variation, all that diversity comes from the sequence of these A's and T's and C's and G's, right? So coming back here, right? The forensics team extracted DNA. They did what they could. There was some damage, and they know that the sequence goes something like this. So A, C, G, T, C, T. Well, with only the application of the Chargaff's law, the complementary base pairing law, we can fill in any gaps that happen to be damaged, right? So we know that if we have an A, its complementary match must be the letter T. If we have a C, we know that C will complementary match with G. G with C, T with A, C with G, T with A. Right? And we would go in there and kind of fill in any question marks with the appropriate base just by the application of the Chargaff's law, right? A with T, C with G. I still haven't quite mastered the little drawing tablet, so I don't know how to necessarily write all the letters here, but if we put this in a real life situation, right? This is the sort of the process that would happen. So DNA is found at the crime scene. The forensic scientists would go in there and completely sequence out that length of DNA, as much DNA as we can, hopefully the entire set of DNA. Then let's say that there was a little bald-headed person that was seen at the area of the crime. Well, we get some suspects that sort of match that description, bald-headed kid, all right? Well, we got 2 bald-headed kids, right? So we extract DNA from both of these individuals. The forensics team is getting lazy on us here, right? They just gave us 1 strand for suspect 1. But we know that if there's a T, that would be matched with an A. C with G, C with G, T with A with T, G with C, A with T, G with C, T with A, C with G. A little bit skewed there, but again, all we're doing is applying Chargaff's law, and we would do the same for Charlie Brown down here. And when we're done, we would then have the complete DNA found at the crime scene. We would have the complete DNA found from suspect 1 and the complete DNA found with suspect 2. And again, this is just a tiny fragment. Again, in a real situation, we'd have 1 million letters that we would sort of match. Now, for this to work, if we're trying to match the DNA of a suspect to DNA at the crime scene, there must be an exact match. It can't be just kind of close. Oh, it almost matched. No, no, no. Is it going to be the exact sequence, right? The exact sequence of that strand. So I'm going to let you analyze this, this slide for a bit. And can you sort of guess? Can you sort of analyze? Can you, hopefully not guess, can you know for sure the answer, right? So members of the jury, right? Would you say that suspect 1 is the guilty individual? Would you say suspect 2 is the guilty individual? Or would you say that neither, maybe they're both innocent, right? So how would we know who is guilty? And there's no hiding from this, right? A person can go and color their hair, shave their head. The person can put on colored contact lenses, right? A person can file down their fingerprints, but we cannot alter our DNA, right? So the DNA, it tells the truth. The DNA has all the information, right? Where do you think these our fingerprints are derived from? These are proteins that are coded for by our DNA. We all have different fingerprints because we all have different sequences of DNA coding for those fingerprints, right? So after analysis here, we think we got an answer. Do we find an exact sequence match somewhere along the, it may not start at the very left. It may start somewhere else, but can you find an exact sequence match anywhere on here? If you did a good job, you would have found an exact sequence match here, right? So we have T, C, C, T, A, G, A, G, T, C. We would have the exact sequence match here, right? So I'm kind of I'm not covering every single little step in the process, but I'm giving you enough information to hopefully visualize how a little sequence of DNA can be used in this sort of forensic science application. And this type of information has sent people to prison. This type of information has acquitted others. This is the kind of stuff that is used today in modern science, FBI, modern forensics. It's a little bit different. We don't apply it the same way, but paternity testing, who's the baby daddy, that kind of stuff. We can apply this type of situation to these ancestry companies, Ancestry.com, 23andMe. They use this sequence data to be able to tell you, your relatives, where your family line is from, what ethnic genes you carry in your DNA, right? So that those processes are different than this particular example. This one, we have to have an exact match because this DNA actually belongs to that person. If we're looking at paternity testing, a child's DNA is not the exact as mom's, not the exact as dad's. Those are, we're looking at percentage similarities when we're doing that. But again, the concept of the A's and T's and C's and G's, Chargaff's laws, those are still applicable. So we know now, and if you knew something about Stewie, little bastard, he was the one that that was going to be guilty, right? But we were open-minded, unbiased. We let the DNA talk for itself, and yes, we found suspect 1 to be the guilty one. So this is again how we would analyze DNA in modern forensics science, right? All right, so let me stop this one. This is a nice short one, so it can load fast. And then I'm gonna address the last part of this particular chapter, the RNA molecule, right? So not a whole lot more to cover today. Stop this here.